So the second topic is aging. And aging, of course, is the common denominator of so many of the diseases we care about now, including, of course, infectious disease. It's no coincidence that COVID hit older populations much more heavily. I mean, it's not inevitable. Obviously, the flu pandemic of 1910, 1911, or 1916, that that hit everybody. But here you see gene network, and we're going to talk about aging now as a common denominator. So I've got many thanks to this work. The work I'm going to be describing is in a Nature paper that is in press, and I hope to have the proofs in the next week or two. But if you just keep your eye out, or I'll send it to Rami when I get a copy of this. This is the title of the paper, Dynamics in Genetics, Dynamics of Genetic and Somatic Trade-offs in Aging and Mortality. And it uses Black 6, but it also uses 3 other strains. There's DBA2J, but then 2 other strains. And I just want to shout out my thanks to many, but in particular Danny Ahrens, who did the bulk of the analysis, and David Ashbrook and Shuhita, who did all the DNA work, and then the many other colleagues who contributed in different ways to this paper. And finally, most of the last authors here made major contributions over 20 years to generating a cohort of about 25,000 mice, all of which had beautiful data on their lifespan. And those are the data we used. We used about 6,400 in this particular study. So what's the question really is, what's modulating rate of aging or aging mortality rates? All of you will be interested in this question. I just list some of the classic questions. A 25-gram mouse lives 800 days. A 25-gram bat lives 3,000 days. Why is that? Humans do pretty well, 30,000 days, but a Greenland shark does 140,000 days. Ouch. I wish, not that I want to be a Greenland shark, but I wish I could live that long. That would be pretty awesome. How can we modulate rates of aging? That's the question. And we would love to do this in humans, but we don't have time enough unless we want to do this over the next 1,000 years. So I want to do this in my lifespan, which means I have to be able to do studies in about 10 years. And the study I'm telling you about, well, there have been 2 studies. They've taken more than 10 years. So here's the first study that we published in Science in 2022, Sex and Age-Dependent Genetics of Longevity in a Heterogeneous Mouse Population. In this study, we took 3,200 animals over here. These animals here, they're called had 3 mice. Instead of having 2 parents, they, like all humans, they have 4 grandparents. And so they're, in that way, an even better model for human genetic diversity than the BXDs. The problem with them is there's only 1 of each. So these don't have identical twins. So there are pros and cons of any model. And so if you then ask, if you start with 3,200 animals up here, let's just call that 100%, and you ask how many of them make it to 6 months, almost all of them make it to 6 months. How many of them make 12 months? Almost all, but a few of the some of the males have died. 18 months, more male mortality, early male mortality. And then the males and the females have about the same mortality rates after about 30 or 32 months. So there was a lot in this paper. Whoops, sorry. You can certainly read it. There's a PubMed ID, but I'm going to show you the update on this paper. And you can see here in this paper, we did as good a job as we could at that point in translating from mouse to human. And we're still working on that aspect. This is the bigger paper, the Nature paper that's in press. And this is based, I mentioned 25,000 of these mice. We then genotyped a total of 6,438 of them And this is the revised curve, starting now at 365 days. Here's a different way to look at it. So at the bottom of this, it's not really a pyramid, but in honor of speaking to you in Cairo, I'm going to call it an age pyramid. At the bottom of the pyramid, we have 6,438 animals. Half are females, roughly, half are males, roughly. And as we go up in age to 600, 800 days, obviously animals die. And you can see the difference in death rates. So the females actually have very little early death. The males have quite a bit of early death starting at 200 days. And that means that if I give you a mouse at 42 days, which is puberty, that's essentially a 13-year-old human in mouse terms. So 42 days is a very young mouse. And I ask, what's the difference in life expectancy when you're 42 days of age or when you're 13 between males and females? You can see there's a huge difference of about 80 days. So the females will live 80 days longer than the males. But however, as the males die early, there is no difference when you get to about 700 days. So the male mice that make it to 700 will live as long or even a little bit longer than the females. So you can see that the mortality rates are tweaked for the two sexes. They're not the same. This is the marker set, the genetic markers we used to study these animals, a total of about 900 markers. We're now switching over to sequence-based genotyping. Here's a map of lifespan for females in red, for males in blue, and for combined males and females in green. Tell you what, on the x-axis, we have all the chromosomes. On the y-axis, we have that minus log p or LOD score. So these values above the orange line here are quite significant. So there's 1 significant, 2 significant, 3, 4, 5, 6, 7, 8, 9, 10, about 12 significant spots in the mouse genome are controlling lifespan, but this is only looking at the animals that made it to at least 1 year of age. So truncation age, 365. So this is now issuing a mouse life insurance policy at 365 days instead of at 13 years of age. So this would be the equivalent of issuing a life insurance policy to a human at about 50 years of age, 45 to 50 years of age. At 365 days, female mice do not reproduce very well at all. So this is basically the menopausal time point for females. And andropause in male mice might be a little bit stretched out, maybe to 500 or 600 days. But a male mouse will never reproduce after 200 to 300 days because the other male mice who are younger will beat it up, essentially. So it won't be competitive. So I want to look at this Vita1A locus. So we're going to look at what does this actually mean? So we have Vita as in lifespan, vitality. So Vita on chromosome 1, our first locus, Vita1A. So let's look at that in detail. What does it mean? It means that there are 4 genotypes animals can have at Vita1A. They can inherit a C and an H, a B and an H, a C and a D, and a B and a D. That's because there are four different grandparents. And these are the combinations of those 4 grandparents that the animals can inherit. And you can see that if you inherit the C and the D or the B and the D, you have a significantly shorter lifespan, minus 20 days, relative to average, which would be 0, compared to CH and BH. And that difference persists until about 900 days, and then everybody converges. And this is, again, sex-combined data. And what that means is that there's been a wave of deaths right here, the rate of change tells you that there is a wave of death of animals that are CHs and CDs and BDs. So whenever you see a rate of change, it means something dramatic happened. And it means that if you're a life insurance issuer, you can tell already by 42 days that you would like to have this, you would like to insure this mouse until about here. And then you would say, I'm going to charge you more for your insurance policy. Fortunately, in America and most places with sensible policies, life insurance companies can't do this, but they could never get this data. So don't worry about this as a future dystopian insurance policy method, because this is highly, highly dependent on your environment. And these animals are raised with scrupulous care for uniform environment. I'm going to skip this plot. This is kind of for geroscientists. But let's look at Vita 1A in a different view. And this is looking at Vita 1A and asking, when do those, I mentioned that there were big swings in death rate. Well, let's look at that, but on age. So I mentioned that there were certain genotypes or alleles that you don't want to have that are going to kill you. So if you're female, so there's a female symbol right here, and you inherit your H allele from your father, you're going to die quite a bit more at 1100 days. In contrast, if you inherit the D allele from your father, you're going to die at 900 days. And this is 0, and you'll notice it's plus in both directions. So this is whatever goes away from 0 means you're going to die more if you inherit that. So you're going to die more if you inherit C or B. Whoops. So these are the first plots that have ever been done like this for any species. This has not even been done for Drosophila or C. elegans or yeast. And I'm not quite sure why. It could have been done for Drosophila, but I think we just had the right data to do it. And you can see that the males and the females are not that different here. On the male side, it's also disadvantageous to have an H if you're above 1100 days. It's disadvantageous to have a D, but it's quite a bit earlier in the males than it is in the females. Anyway, so that is what we're looking at. We would love to know what is causing these sex differences and the sex similarity. I'm going to skip this in the interest of time because we're getting close to the hour. But I just want you to know, as it says here, the sex differences are impressive. The sex differences are impressive. The sex differences are impressive. If you look at these are females and these are the males, you will see very little similarity. And this is the same species. You know, it's not like we're talking about rats and mice. Same species, raised in the same environment, just the different sexes and very little overlap. So in this study in Nature, we've defined 29 modulators of mortality. And the question I just want to move quickly to is, do these genetic loci interact? And remember, the title of this talk was Complex Genetics. So I'm sorry, but life is complicated, and it's more complicated than I can even describe. But let me show you how complicated it gets. We've got 29 modulators of when you die and probably what you die of. So some of these will be heart disease. Many of them will be cancers in the case of mice. Probably most of them are cancers in the case of mice. Some of them are male specific. Some of them are female specific. But do they collaborate? And the answer is the loci that are female specific and the loci or mortality controllers that are female and the mortality controllers are males have almost nothing in common. So what we're doing here is we're looking at pairs of these loci, Vita 1A and Vita 2A, and we're asking, do they work together in some interesting nonlinear way, like a receptor and a ligand, or are they independent? They don't talk to each other, they just do their own thing. And the answer is there's a lot of independence down here in quadrant 3, where there's nothing much going on here. It's shared by males and females. Up here are some highly significant interactions between 3A and 5A, 1B and 9A, 1C and 2B. All of this is a significant interaction, but you'll notice these are all significant in females. These are all significant in the males, and there's not a single common interaction, epistatic interaction here. So again, males and females are like two species for the price of one. There's a great deal of sexual genetic diplomacy to achieve reproductive aims. Now, these are mice. They're very different than humans, so I don't know to what extent this will apply, but obviously males and females of any species have a different idea of what goodness is, what makes them fit, what gets into the next generation. They have to cooperate at some point, but they also have very different individual goals. And here you see it very clearly at the genetic level. And just another example of this, which I will skip you can read the paper in a few weeks, I hope. And I'm just going to skip this and give you a very short overview of the last part, which is Alzheimer's disease. And the good news for this is I can go very quickly because the paper just came out 2 weeks ago in Nature Neuroscience. So if you're interested in this topic, there's just tons of good data. And I'm just really going to take you through some of the figures from this paper. But the question is, how can we use our BXD mouse models to study Alzheimer's disease? Some of the classic questions: can we actually translate from mouse models to human models of Alzheimer's disease? Yes. Can we figure out what parts of the brain are hardest hit by Alzheimer's? Yes. Every one of these questions, there's a yes answer. Some we're not quite there yet. The power is too low, but we're getting there. In the early part of this millennium, I literally dissected mouse brains. I would take a mouse brain out of the cranium and I would chop out specific parts: the hippocampus, the cerebellum, the olfactory bulb, the the whole cortex, the thalamus, the hypothalamus. Now we can do it digitally using magnetic resonance imaging and light sheet microscopy. And that's what we show we showed that initially back in 2020 in a neuroimaging paper, but we did it in the context of Alzheimer's in this paper in Nature Neuroscience. And I'll send that to Rami also. Actually, I already did send it to Rami, so he has that if you're interested in Alzheimer's disease.
The papers were shared in the reminder email you all got before the webinar, so you have it in your inbox, and we can share also later in a folder.
Perfect. So this just shows you what digital dissection looks like. This is the front of the mouse brain, the back of the mouse brain. Instead of getting a scalpel out and saying, where's the dorsal striatum? So this is the part of the brain that's messed up in Huntington and Parkinson's disease. I can actually digitally dissect it with using MRI and methods. And all credit to G. Alan Johnson at Duke. So this is entirely his imaging workflow. It's an amazing workflow. He's been doing it for about 50 years at Duke, and he's the best small animal neuroimager on the world, on this planet. And I'm just delighted to be able to collaborate with him and his extraordinary team. But he's now adopted some of the genetic methods that we've used, and now he has applied them to over 1,000 brains. So this kind of quality of digital dissection is now in scope. I just wanted to show you. So these are different levels. Ideally, I would zoom in on this, but I don't think I have time. I don't have time to go over the cross, but basically we're mixing BXDs with a 5x FAD model. So it's a very well-known mouse model of Alzheimer's disease. And here is just showing you how much difference we see. So all of these are regions of interest, chunks of the brain that have significant differences between the cases that have the Alzheimer's disease allele and the controls. So over 50% of the regions that we see have big differences. And what's fascinating, if you look over here on the far right, everything that's pink actually gets bigger in Alzheimer's disease, and everything that's blue gets smaller. And so the absolute brain size doesn't change at all. 4 brain regions that are used for executive function swell, and we think that swelling is pathological. We think it's because of inflammatory tone. The tissue is very unhappy, it's swelling, microglia are climbing in there, getting just everything inflamed and reducing function in this region. This reduction in volume here could also be dysfunction.
Hi, Rob, I did something. I'm sorry to interrupt, but I finally could share your PDF. Does it look clear for everybody now? Can you see it?
Can I don't see anything other than my.
Oh, you can't see it. Okay, I'm sorry. So
okay. I just see my screen, Rami. I'm just stuck with my screen.
No, because it's much clearer if you can see the shared screen. You can ask me to move the slides, or otherwise we can go back to your.
You want? I don't know what to do.
Your teams. I'm sorry for this interruption, but it may be.
I see something here.
The slide is very interesting. So,
what would you like me to do?
If you can check the Teams, like the Teams application,
check
Teams app.
It looks good to me.
So here on Teams, I share the PDF at the slide.
Oh,
that's right.
Okay, it says that's much better. Okay, and now there's a sign in. Well, okay. So if you want to just run it, Rami, I can just tell you what to do next.
But can you see it? If you see it, then I can do that.
Yeah, I can see it. Yeah.
All right. Yeah. So now,
sorry,
now I am the. No, it's fine. It's something out of our hand. But now I have the delineation slide.
Okay.
And you can tell me to move to the next. Yeah.
Okay. So this is just a blow up of what I mentioned, that the parts of the brain in pink get bigger and the parts in the brain in blue get smaller. This is a huge eye opener to the field, because for the last 10 years, people have been saying there's very little difference. The mouse model of Alzheimer's is a lousy model because it doesn't degrade function very much, and there's no structural change. Wrong. There's huge structural change. It just is relatively small on the plus or minus 10%, but that's enough to screw up function. And if you go to the next slide, Rami, we show some of the kind of consequences. Now we're going to look at the light sheet microscopy. We take the same brain that we've imaged with the MR, we take the brain out, we do light sheet microscopy at much higher resolution so we can count cells. Next slide. And this just shows that we see a lot of A beta 42 in these brains. All the little red dots are the cases that have the transgene, and A beta levels are high. The controls are the blue points. They don't have much A beta. Next slide. Shows the volume difference. Now, these volume differences don't look like much, but they're significant in quite a few cases. So that's what we were able to pick up using these extraordinary MR methods that have very precise abilities to delineate structures. And the next slide just shows that some of the neuron numbers are reduced.
Sorry. Yeah.
Okay, so this is a slide just showing we use an antibody that's generally selective for neurons. And in general, the numbers are either not perturbed greatly or are slightly lower. but only very slightly. And you can see there are two structures there with asterisks. The VCS, that's the visual cortex secondary, which it actually has higher counts in the transgenic mice, which we think is due to lack of specificity of the antibody in the transgenic mice. We think that the transgenic mouse brain is so unhappy that the antigens don't know what to do anymore, and the antibodies don't know what to do. So next slide, we're getting very close to the end. I'm sorry, folks, we're a little bit over here. So
no, it's fine. So this is the right slide with the correlation.
Okay. Yeah, so this is a slide that just shows that that there is a difference between cases and controls. At the what you're looking at there is volume of the cortex on the y-axis, and essentially what their memory function is. And a downward slope means their memory function is not good, and an upward slope means there's not much change or a little bit of an improvement. And you can see a big difference there between the cases and the controls. So keep going, Rami. So conclusions so far, I think I've given you the conclusions. It's looking great. This paper in Nature Neuroscience used only 57 cases. We have 10 times more than 10 times more. So I think we'll have some more actionable results over the next couple of years.
Do you need volunteers to help?
Yeah. Okay, so I think just go to the last thank you slide or whatever I have there. Skip that. Oh, it's all open data. What I've learned, that I think everybody there might get a kick out of that later, but just keep going. And just my thanks to many individuals in the lab. Next slide. Yeah, so these are all the people that have contributed most consistently over a long time. There are lots of other individuals, but I just want to thank them. All right, thank you very much. And it is 8:10 in Cairo.
It's fine, Rob. We are enjoying it. So if you can tolerate the 10-15 minutes of questions.
I can tolerate another hour or two.